Okay, I think it's time to start. So just a reminder, it's not a requirement, but in case you're interested, it could be a really good seminar. Bethany Rader in Andrews 101 at 4 o'clock this afternoon. And there's actually free coffee and cookies if you come 15 minutes early and they haven't all been eaten up. So on Wednesday, we talked about transcription and how it begins. And in the next section, which we're going to start either later today or probably next week, we're going to talk about the regulation of transcription and the regulation of the initiation of transcription. But one of the things that I think sometimes gets neglected is the discussion of how transcription stops. Because transcription is an energy-intensive process, and so bacteria and eukaryotic cells, for that matter, have invested means to make certain that they don't waste a lot of energy transcribing DNA into messenger RNA when they don't need it. So in bacteria, there are two forms or two flavors to transcriptional termination. So these are known as factor-dependent and factor-independent. So these are the two general classifications. We're going to first talk about factor-independent transcriptional termination, which is sometimes referred to as intrinsic transcription termination. Now, intrinsic tells you something. It tells you that the signal for terminating RNA polymerase activity lies within the nucleic acids being transcribed. And so the intrinsic terminator in DNA, which will stop transcription, has an architecture like this. So shown here in this top, here's the DNA. There's an error in this textbook that I stole this from. Notice that the DNA on the top strand is listed 5' to 5', and the bottom is 3' to 3'. Yeah, pretty big error, right? But I still use this one even though it has an obvious error where this should be a 3' here and this should be a 5' here. Because of the color coding, and I like bright colors. So you have an architecture where you have what's known as an inverted repeat. So notice that the sequence shown in red here, 5' to 3', TCG, ACTG, has an inverted repeat. So for an inverted repeat, you have to look at the other strand downstream, because remember the other strand is 5' to 3', TCG, ACTG. It's the same sequence here, but it's on the other strand. So a really common mistake, especially on exams by Biology 306 students, is the failure to recognize what an inverted repeat is. Because what people will think is that if the sequence on that same strand is an inverted repeat, but it's obviously not. Now, the reason why inverted repeats are important is not at the DNA level. Inverted repeats exert their effect, in this particular case, at the level of the messenger RNA. So here is one part of the architecture of an intrinsic terminator. Here's the left half of an inverted repeat. Here's the right half of the inverted repeat. And then the other really critical component of the architecture of this intrinsic terminator factor is this poly-T tract immediately downstream of the second half of that inverted repeat. Is generally at least 6 or 7 thymines in a row. Now again, at the DNA level, this is just a unremarkable piece of DNA. But as RNA polymerase transcribes this, making a messenger RNA copy of that bottom strand, you can see the effects of these inverted repeats. So recognizing that this UCGA was made as a RNA copy of this top strand. But because RNA is single-stranded, it has the ability to base pair internally. So it has the ability, and does this quite profoundly, to fold back on itself based on base pairing. So by having an inverted repeat, this builds in a structure known as a stem loop that can form in the messenger RNA. Because you have base pairing between G and C, between uracil and adenine. Remember that at the RNA level, base pairing, is the same as that DNA. Uracil substituting for thymine. So you get this very stable stem with base pairing between these complementary ribonucleotides, and you have this loop structure here. And then it's followed by a polyuracil tract, the RNA equivalent of the poly-T tract. This stem loop structure, when it's followed by a polyuracil tract, is a signal to bacterial RNA polymerase to halt and to dissociate and to fall off. The way that this architecture at the messenger RNA level works to cause the RNA polymerase to fall off is actually shown, or well, I've gotten ahead of my bullets here, but that stem loop will actually destabilize RNA polymerase. So RNA polymerase's ability to hold on to the template DNA strand is going to be negatively affected by the formation of this intrinsic terminator or stem loop structure. And that's what's kind of shown in this cartoon here. So here, this football-shaped red sphere, or rugby shape, actually, probably closer than a football. Sorry, Matt. You see that this RNA has now, the RNA polymerase has transcribed through the messenger RNA, through the inverted repeat, so it's formed this stem loop. Now, imagine that before that stem loop was formed, this portion of the messenger RNA was base paired with the template DNA, just as these U's are base paired with the template A's. The only thing that holds RNA polymerase onto the template DNA is the hydrogen bonds between the template and the nascent strand of messenger RNA, the messenger RNA that's being made. So anything that decreases the hydrogen bonding between template and messenger RNA tends to destabilize that enzyme DNA structure. So what happens is that when that inverted repeat is transcribed into messenger RNA, the messenger RNA will actually form that stem spontaneously. Because RNA-RNA base pairing is favored over RNA-DNA base pairing. So this stem loop will form when you have an inverted repeat, and then if that second half of the stem is followed by a poly-U tail, remembering the difference between a GC base pair where there are 3 hydrogen bonds and an AT, or in this case a uracil adenine base pair where there's only 2 hydrogen bonds, this is inherently unstable. It has very few hydrogen bonds. And so that's going to promote the dissociation of that complex. Notice that this RNA polymerase is not really shown here in subunits, but remember that the RNA polymerase no longer has that sigma subunit. The sigma or specificity subunit that bound to the promoter is gone at this point. So really, you can kind of picture this, or my analogy in my mind is a train going down the railroad tracks. Once you get that train moving, it doesn't take very much energy. There's not much friction tending to stop that train. It's the same thing with RNA polymerase. Once sigma is gone, it can move easily down the DNA because it is almost, think of it as frictionless, because the interaction is only between the nascent messenger RNA and the template DNA. And when a stem loop forms, it falls off. So if that picture doesn't resonate with you, hopefully this one will. So hopefully, I love pictures. I'm a very visual learner, and I hope you guys are too. So I put in a separate picture. So here is at the DNA level, another example of an inverted repeat. And just, make note of this. I can guarantee you that there will be questions on the next exam where I'll show you several DNA sequences and ask you to pick out the inverted repeat. So, and they will be trick questions because I will make it such that, so here, AGCCC, if I put on this top strand coming backwards, AGCCC, even though I'm telling you right now that's not an inverted repeat, 20% of the class will still pick the wrong answer. So the other half of the inverted repeat is on the bottom strand. So you see AGCCC is the same as AGCC here. It's not showing up very well here, but on the opposite strand, these are T's. So when that gets transcribed, so I like this picture better because it shows here that the first half of that inverted repeat has been transcribed into messenger RNA, shown in the light green. And now you have all of this base pairing between the nascent messenger RNA and the template. This is that open complex. We talked about when RNA polymerase initiates transcription, it melts the DNA locally to form this bubble or open complex. And you can see that there are, 15 or 16 nucleotides base pairing the messenger RNA to the template. That's the only thing that's holding RNA polymerase on. The one thing I don't like about this picture is it's not obvious that this is going on within RNA polymerase. So as you're looking at this picture, imagine that RNA polymerase is surrounding this entire open complex circle and synthesizing nucleotides, putting on nucleotides on this 3' end. But now you have, or the bacterium RNA polymerase has created the inverted repeat. It's transcribed both of them. So now this is going to form spontaneously because the RNA-RNA base pairing is more stable than the RNA-DNA hybrid, shown here in step 2. What that does is that robs RNA polymerase of that level of stability. All of the hydrogen bonds between these G's and C's are now gone, and it's just the weak hydrogen bonds or the reduced number of hydrogen bonds between A and U here. So this is going to fall apart. Transcription is going to stop right there. So this is a very efficient way that bacteria use to stop transcription. It doesn't require any other proteins. There are no other transcription or termination factors involved. It's intrinsic. It's built into the DNA. Now, that's in contrast to the other flavor of transcriptional termination. So that we first talked about the intrinsic or factor independent. This is factor dependent. So the transcriptional terminator here is not intrinsic to the DNA. It requires a protein factor externally. The most common one in bacteria is a protein called Rho. There are some others that are involved in this. And don't confuse Rho with the eukaryotic Rho protein. Those of you who have or are taking cell biology, Rho is a completely different thing in cell biology. So here, Rho is a protein that has the capability of binding to messenger RNA. So I don't know why this projects up so high on this, but here is, let me come down to this next one. Here is RNA polymerase with the beta and beta prime shown in brown and green, the 2 alpha subunits. And this actually shows that little omega subunit here. But sigma is gone. So this RNA polymerase is moving down the DNA. It's causing this local melting. And the nascent messenger RNA is shown here in red. So as that RNA emerges from the RNA polymerase, it's going to be bound by this protein Rho. Rho is a pretty sizable protein. And once it binds, it forms hexamers. So it forms a multimer of itself. And it literally chases. It has the ability, by hydrolyzing ATP as its energy source, it has the ability to chase RNA polymerase down the DNA. And actually, Rho moves faster down the RNA polymerase, or down up that messenger RNA, than RNA polymerase goes down the DNA. So this is not a fair not a fair fight. It's not a fair race. This Rho complex is going to catch RNA polymerase, and when it does, it interacts with RNA polymerase and kicks it off. So how is it that the bacterium makes certain that Rho doesn't catch RNA polymerase before it's finished transcribing what it needs to transcribe? That really has to do with the fact that we're doing this in prokaryotic cells. So I'm going to get on a soapbox for a minute. You've all learned that one of the primary differences between prokaryotic cells and eukaryotic cells is that prokaryotic cells don't have a nucleus. And that's true. That's very true. But now, as scientists, you need to take those trivial pursuit answer types of questions and think about, what does that mean for the biology of the cells? Because the absence of a nucleus in bacteria allows for a very different biology to occur than can occur in a eukaryotic cell. In bacterial cells and other prokaryotic cells, translation and transcription occur at the same time in the same compartment of the cell. In eukaryotic cells, in yours and mine, transcription is occurring within the nucleus. Translation doesn't occur until that messenger RNA is transported across the nuclear membrane into the cytoplasm. So those two processes are separated spatially and temporally. But in bacteria, that's not the case. And so bacteria have taken advantage of that seemingly trivial difference. Well, it's not a trivial difference, but you might think of it in terms of, oh, that will be an exam question, the difference between a prokaryote and a eukaryote. That's what I mean by trivial. But the bacteria have taken advantage of the fact that they're doing transcription and translation at the same time, because the ribosomes that are translating the messenger RNA have the ability to block Rho. They can literally slow Rho down from catching up to RNA polymerase. So I like to think, I like analogies. And I hate the fact that people in Virginia, and I'm sorry for Virginia residents, drive in the left-hand lane on the highway, even if they're not passing a car, they won't get the hell out of your way. Oh, I get so, I'm a very laid-back person, but road rage is my big problem. And I get so mad at people for doing this. And so I always think about, I think of them as a ribosome, and I'm Rho, trying to get home. Because what happens is that as that messenger RNA is emerging from RNA polymerase, it's bound by a ribosome, and it's also bound by Rho. So the ribosome and Rho are both chasing the RNA polymerase. The ribosome is going to chug along, making protein. I should have drawn protein coming out of this, until it reaches the stop codon, which I put this little red star in. So at a stop codon, ribosomes are programmed to disassociate and fall off. So this is where the ribosome takes an exit from Highway 64 and gets out of my way. And now Rho can catch up to RNA polymerase and kick it off. And this makes sense because at this point, the gene has ended. If this is a protein encoding gene, here's the stop codon. The ribosome disassociates, and now Rho can catch up to RNA polymerase. So it doesn't bother to transcribe a lot of needless DNA into messenger RNA. It transcribes only a short distance beyond that stop codon. So Rho catches up, it forms a stem loop in the messenger RNA, destabilizes it, breaks the whole thing apart. So this is the other mechanism by which bacteria stop transcription. So transcriptional termination is almost as important, if not as important. Well, no, I shouldn't say almost. It is as important as transcription initiation. You have to be able to stop transcription at the right point. Okay, so we're going to move on now and talk a little bit. I'm trying to get into some things about mutations in bacterial DNA. I want to talk about DNA repair, DNA mutations. But just by way of a quick review, because I know that you've all had this before in intro bio classes, but some of you, it's been longer since you've taken intro bio. So this is a codon table. So remember that at the DNA level or at the messenger RNA level, proteins are encoded in codons or 3 nucleotides. So there are 64 possible codons, 61 of which code for amino acids, 3 of which are stop codons. So you can, when ribosomes encounter UGA, UAG, or UAA, ribosomes fall off. Now you've probably all learned, and part of the reason why I'm putting this back up as review is that if you didn't learn this in a microbiology class, you learned only partially correctly that AUG, the methionine codon, the single one methionine codon, is the start codon for proteins. And that's true in eukaryotic genes. Every protein encoding gene in a eukaryotic cell always starts with an AUG codon. And it's true about 2/3 of the time in bacteria. So bacteria very frequently use AUG, but they also use GUG, or at the DNA level, obviously GTG, which normally encodes a valine. But yet, bacteria, even though they can use GUG, and to a lesser extent UUG, which normally encodes a leucine, they still always put a methionine in there. And the way you can remember these 3 codons is, this is kind of the opposite of the wobble position. So you all learned, for instance, that the redundancy in codons, in the codon um, table here means that that third position, so if you look at proline, so proline can be encoded by 4 different codons, right? They are all GC, and then any one of the 4 nucleotides in that third position, or the wobble position, will still allow proline to go in there. It's kind of the opposite with bacteria and the start codon. So they'll usually use AUG, sometimes they use GUG, and sometimes they use UUG for leucine. So they can vary that first position. But they still always put in methionine. So I don't really know truly the significance of why they use 3 different codons, but the thing that's important to remember is that bacteria, unlike archaea and unlike our cells, always put in that first methionine, not regular methionine, not the normal methionine that we all use in our protein. They put in a modified form, N-formylmethionine, or FMET. Now, I kind of misspoke a little bit because I said that eukaryotic cells, yours and mine, don't use FMET for the beginning of their proteins. There is an exception to that. In our ribosomes, in our mitochondria. Mitochondria, remember, are domesticated bacteria, and they've lost a lot of the characteristics of being a bacteria. They no longer have lipopolysaccharide, for instance. They don't have peptidoglycan, for instance. But one of the things that mitochondria have retained, which is indicative of their ancestry as a bacterium, is that their proteins always begin with N-formylmethionine. So this is somewhat of a trivial pursuit matter. And I was chastising myself last night as I was thinking about this lecture. I always tell you, like I just did with the whole idea of bacteria not having a nucleus as opposed to eukaryotes. Remember, what is the effect, or what does it allow the biology to do, this absence of the nucleus? And I was thinking to myself, it's like, well, I always present to students that bacteria use N-formylmethionine at the beginning of their proteins, but I never tell people why it's important. And then I realized I don't know why it's important that bacteria use these. So these next 2 slides are my way of updating the lecture for next year. You guys don't need to know these next 2 slides. They're not in your set. So I just put these in this morning because it was only this morning that I found out why it is that it's important that bacteria use N-formylmethionine. So we're not going to spend a lot of time on this. But basically what happens is that as a bacterial ribosome translates messenger RNA and puts that N-formylmethionine on it, there is a process associated with the ribosome that removes that formyl group from the methionine. And in some cases, actually removes the entire methionine. So in most cases, even though bacteria, so this was the big mystery for the longest time. It's not only weird that bacteria use N-formylmethionine, after they use it, they cleave it off. So why the hell did they ever use it to begin with? So that's what's shown here and in the next slide. So we know that under normal conditions, as the protein emerges from this ribosome with an N-formylmethionine on its amino terminal end, there is a protein deformylase, that's what PDF stands for, activity that removes the formylmethionine. So if this is a normal protein, perfect. But what the N-formylmethionine does is it's a safety check to make sure that the protein is actually forming normally. Because if something happens, let's say the bacterium is growing at an abnormal temperature, slightly high. So now as that same protein emerges, if it misfolds, if it doesn't fold into the proper secondary structure, that N-formylmethionine is no longer put into a position where it can be accessed by this PDF activity. And so the formyl group, the formylmethionine, remains on it. This is actually a tag that bacteria use to determine that proteins are defective. They have been misfolded. And that N-formylmethionine will actually allow the protein to be targeted to a degradation pathway, kind of similar to the proteasomes in eukaryotic cells. So this is the significance of the N-formyl methionine. And as it turns out, this is very similar to something that's happening in our cells. We do this as well, but we use an acetyl group instead of a formyl group. So it has a similarity. So like I said, this was just because I knew you wouldn't be able to sleep tonight if you didn't know what the N-formyl methionine, but in terms of like studying for the exam, don't pay any attention to this part of the lecture. This part, however, might be on an exam. Because part of the reason why I give that little bit of an introduction is because what I want to talk about next in class is our mutations. So, and again, we're going to talk a little bit about of a review of mutations, but I'm also interested in you remembering the difference between a transition and a transversion mutation. I really don't care about that. What I want to be able to show you is how bacteria have evolved to take advantage of mutations in order to adapt to changes in their environment. So in terms of mutations, bacteria have a mutation rate not much different than ours. So between 10 to the minus 7 and 10 to the minus 11 per base pair. So what does that mean? What that really means is that this 10 to the minus 7 would mean that you have to replicate a DNA position, any one nucleotide position, 10 million times before, on average, you would make some kind of a mutation at that spot. So pretty damn fidelitous. And what's 10 to the minus 11? Like 100 billion? So you'd have to replicate 100 billion times before, on average, you would find one error at that nucleotide. So very fidelitous, very similar to ours. And now, if you kind of take this to its logical extension, if the typical prokaryotic gene, bacterial gene, is about 1,000 base pairs in length, and taking the spontaneous mutation rate that I just gave you here, that would mean that in any one gene, you would have to transcribe or replicate that gene 10,000 times before, on average, you would have a mutation in that gene. But so this is the beauty of microbiology. Because the bugs are so small, they grow so rapidly, and to such high numbers, you can easily get a population much in excess of 10 to the 8 cells within a few minutes in the laboratory. And you know that within that population, there lies a mutant in your protein gene. It's just now the genetic challenge becomes how the hell do you fish it out if you're mutant hunting. But because bacteria numbers are so large and grow so fast, this is why from a genetics point of view, an experimental genetics point of view, it's so great to work with microbes. But these mutations occur spontaneously for the most part during replication. So we're not going to really get into the fact that things like chemical or physical agents, like ionizing radiation, can cause DNA. We're going to touch on that a little bit with DNA repair mechanisms. But for the most part, and I think you already know this, that most mutations that occur in our genomes and in any genomes occurs just randomly during the replication of the chromosome. And then the other major source of mutations, and we're going to touch on this, is during DNA repair. So when DNA is damaged by a chemical or by radiation, we and bacteria have mechanisms to repair that DNA. And sometimes those repair mechanisms might allow the cell to live, but they make errors. And the errors are just the price of survival. Now, again, I'm not really interested in you knowing what a transition or a transversion mutation is. The only ones that I'm really interested in you having an understanding of are the difference between synonymous mutations and non-synonymous mutations. So this is more of an evolutionary term. So synonymous mutations are also known as silent mutations. That's where you can change a triplet code and it gives you the same amino acid. So for instance, AGG is the codon for arginine. If you mutate the first nucleotide to a cytosine, it still encodes for arginine. You can do some selection based on codon preference, but for the most part, the protein that would be produced as a result of this mutation would be just the same as wild type. So this would be a synonymous mutation or silent mutation. More interesting, or more impactful, I should say, are non-synonymous mutations. So in non-synonymous mutations, you have a change in the nucleic acid sequence that results in a change in the amino acid sequence. So there's just 3 different classifications of this, and again, I'm not interested in you recognizing the difference in these classifications, but you can have a mutation that changes a lysine to an arginine. And those of you in biochemistry realize, lysine or arginine. These are both similar amino acids, so probably wouldn't have a major effect on the protein function. Or you can have ones like the missense mutation, where you get a very different amino acid, or you can have a mutation known as a nonsense mutation, where you mutate a codon that should normally encode, in this case, glutamine, and turn it into a stop codon or a nonsense codon. So those are just the different types of non-synonymous mutations. But the mutation that I'm most interested in, and this is for me personally, and so because I'm interested in insertion and deletion mutants more than any of these other mutants, that means you are more interested in insertion and deletion mutants, at least for the rest of this class. So here is a hypothetical mutation that can occur, but very rarely. So here is a DNA sequence. Here's the wild type strand shown on the left. And here it is on the right, where you can see this box. There's been the insertion of this base C with the concomitant G on the other side. This is a very rare mutation. This particular sequence, as you're looking at it here, would probably replicate 1 billion times before you would either randomly insert a nucleotide or delete a nucleotide. What would occur much more frequently would be changing one nucleotide, say from a G to a C or something like that, changing a nucleotide. But inserting or deleting in just a normal piece of DNA, extraordinarily rare. You're going to see a situation where it becomes less rare in a moment. But in this, from an older textbook, you see the result of this mutation. So here is the corresponding messenger RNA, and the codons are shown. So this piece of DNA would be transcribed into this messenger RNA, which would be translated by a ribosome into methionine, proline, tyrosine, tryptophan. But if you do this mutation where you insert a C, you change the reading frame. So now you still make methionine, you still make proline, but because the codon has changed, instead of what was UAC, it's GUA, which will give you valine, and then leucine. So very different amino acids. So these kind of mutations, even though they're very rare, where you either insert or delete a nucleotide, have a very profound effect at the protein level. They literally will destroy the rest of the protein after that, because none of this normal amino acids will be produced. But as I mentioned, in a sequence that looks like this insertion, extraordinarily rare. But these insertions and deletions are really critical for something that I find fascinating, and it's widespread among bacteria as a means to adapt to changing environments, both inside hosts like you and I, but in the environment. So we're going to look at mutations like this, insertion or deletion, that are going to drive two really critical acclimations or adaptations that bacteria do, called phase variation in one case and antigenic variation in the other case. We're going to deal first today, for the rest of this lecture anyway, with phase variation. So in phase variation, basically what happens is that you have a gene for a protein that can exist in two phases. Phase on, where it can be and is expressed as a protein, and that same gene in another cell in the population can exist in phase off. So it has a mutation in it that has caused a frame shift that makes the protein incapable of being synthesized. So now what this does for the bacterial population is that it allows that population to have basically two kinds within it. And so if natural selection favors the lack of production of that protein, the cells that don't produce the protein will do very well. But the cells that don't produce the protein won't. And then if the conditions change, if the environment changes, the bacteria just shifts. So natural selection favors the other form. So this is actually going to occur, this phase variation or on-off, is going to occur by an insertion or deletion mechanism called slip strand mispairing. We're going to come back to antigenic variation later. For the moment, I want to talk about the mutational mechanism called slip strand mispairing, which drives phase variation. So I don't love this example, but it shows it nicely. Because the DNA that I showed you before that had the insertion in it of one nucleotide, I said was a very rare mutation. And it is. Incredibly rare. 1 in a billion, perhaps. You can increase the frequency of an insertion or deletion mutation if the DNA is repetitive. So that's what's being shown here. So here you are replicating, we're looking at DNA replication across a template strand where you have this 3 nucleotide sequence repeated in tandem. GTC, GTC, G. So there are 6 GTC, think of them as a codon. And so replication is occurring, making CAG, CAG, CAG. This process of slip strand mispairing occurs at repetitive DNA, and only at repetitive DNA. Because what can happen is that, and this isn't incredibly frequent, but it's much more frequent than a normal mutation rate, is that the newly synthesized strand of DNA, shown here in blue, can come transiently disassociated from the template. And then because it's repetitive, the 3' end can either reassociate with the DNA that it just replicated, or it can form a loop, as is shown here, and now perfectly base pair again with the template, and continue transcription in this case. So now, the daughter cell that would be created here has got 7 tandem CAG codons. So you notice that number 2 is shown here twice. You can also, during the same process, that loop that forms doesn't have to be in the nascent strand or the newly synthesized strand. The loop can occur in the parental strand. And when that happens, now the newly synthesized daughter strand has got only 5 CAGs. So this repetitive DNA increases the mutation rate tremendously. Maybe 10-fold, maybe 100-fold. That still makes it very rare, right? So if a high mutation rate in bacteria is 10 to the minus 7, and this is 100-fold more, it's still 10 to the minus 5. So most of the time, when bacteria replicate this repetitive DNA, they do it just fine. And the daughter cell gets 6 CAG codons. But at a higher frequency than normal, than in non-repetitive DNA, some cells are getting out of there with 1 more or 1 less of these codons. Now, the reason why I don't love this particular example is this wouldn't have a profound change in the protein. Because here you would just have, CAG is a glutamine codon. I had to look that up. I wanted to sound impressive, like I knew what the whole 64 codons coded for. I don't. But I looked it up before class. CAG is a glutamine codon. So normally this cell would have 6 tandem glutamines in a row in that protein. This one would have 7, this one would have 5. But the rest of the protein downstream from that would be perfectly fine, just like normal. So there's a pretty good chance that just adding or deleting 1 glutamine within a stretch of glutamines of that primary structure maybe doesn't have a very big effect on that protein. So I use this slide because it kind of illustrates slip strand mispairing. But the bad thing about it is I think it doesn't illustrate how profound the effects of this can occur. Riley?
Does this also happen in eukaryotes?
Yep.
Aggregates are responsible for causing these things.
Yes, so this does occur in eukaryotes as well.
Okay, it's just the same process.
Yep, exact same process, right. Matter of fact, I think I stole this from a eukaryotic cell biology paper or book. So here again, with a little bit less dramatic nature to the repetitive sequence, you can see it here as these T's. So now the repeat is not in multiples of 3, but it's in multiples of 1. So you can have, as this repetitive DNA is made, you can get slippage in the new strand where you form the loop here, or you can get it so that the loop is formed in the parental strand. But now you have an increase in that T length of 1, or a decrease in the T length of 1. That's going to have a much more profound effect on the protein because it's not in multiples of 3. Multiples of 3 are codons. So that if you increase or decrease by 3, you just add or delete an amino acid. But if you add in one nucleotide or delete one nucleotide, or the same thing if it's two. I'm going to show you an example of a repeat that has a dinucleotide repeat. Either one of those will change the reading frame for the rest of the protein, and so the rest of the protein is destroyed for the most part. So I'm going to use an example from my own lab. So this is a bacterial protein that in Helicobacter pylori that encodes an outer membrane protein that's important in the virulence of this bacterium. So SAB-A, shown here, encodes an outer membrane protein, similar to the porins that we talked about earlier, that allows the bacterium to adhere only to gastric cells or stomach cells when those cells are inflamed. So there's something that occurs during the inflammation of the human stomach that causes the protein expression of the eukaryotic cells in your stomach to change so that they begin to express a protein that they don't normally express. And when they do that, the protein product of SAB-A can help to bind to it. So it helps the bacterium survive during inflammation. But if the stomach is not inflamed yet, there is no advantage and probably a disadvantage to expressing this protein. So the way that this SAB-A protein gene has evolved to make certain that any population of this bacterium within anybody's stomach is always going to consist of some cells that express SAB-A and some that don't express SAB-A. That ensures the fact that regardless of whether the stomach is inflamed or not inflamed, somebody can take advantage of that situation. Somebody can survive. And then if the inflammation goes away, you can switch the population dynamics back to one that's not expressing it. So here, near the 5' end of this gene, I've shown it here in purple, is a dinucleotide repeat of pyrimidines, CT. CT1, CT2, 3, 4, and 5. In this particular arrangement, here's the start codon. If you look at the codons, it would make a methionine, a leucine, a serine, a leucine, and then it gets a nonsense codon or stop codon. This is all at the beginning of the gene. The rest of this protein is completely out of frame and cannot be translated. So a bacterium that has this arrangement of its CT tract is phased off. It doesn't produce the protein. But as this bacterium replicates more frequently than a random piece of sequence, there's going to be a mutation occur by that slip-stranded parent, which in this case shown here leads to an increase in the CT tract length. It could also take out a CT as well. But in this case that I'm showing you here, it has added one CT. And so now you see that what was a stop codon here in the above one, this TAA, is now here. But it's no longer in frame to be a stop codon. So you get methionine, leucine, serine, asparagine, and on and on and on, until you get to the actual stop codon more than 1,000 codons downstream. This bacterium is now phase on and is expressing the full-length protein. So now within that population, you have both phases going on. And so depending on what the environment selects for, you can never get rid of the bacterium. This is part of the reason why Helicobacter pylori, so I'm getting off topic a little bit, Helicobacter pylori is one of the most unusual bacterial pathogens in that once you're infected with this, you are infected for life, unless you're treated with specific antibiotics. Most other bacteria like Salmonella, Shigella that infect the gastrointestinal tract, if you don't die from the infection, you get rid of it, it's gone, and you're immune to it. Helicobacter pylori can stay for decades, for a lifetime of a host. And part of the ability to do that is that they can phase vary between the expression of not just this protein, but dozens of other proteins. So there's nothing that the host, us, can throw at the bacterium that it can't adapt to at some level. So that's why I say this phase variation, which results in translational phase variation, is so important. Because by translational phase variation, I mean you can either produce a protein, as on the bottom, or not, as on the top. Now, 6 minutes left. The next topic that we're going to go into is another form of variation. But rather than phase variation, where you have one protein that's either expressed or not expressed, in antigenic variation, which is a form of this phase variation, you can express one of two different proteins. but you express one or the other, never both, never neither. So what this does is this allows, for instance, Salmonella has the ability to vary which one of its flagellar proteins it produces in a host. So if a host has an immune response against one type of flagella, the bacteria that are expressing that kind of flagella are killed. But any bacteria that are expressing the alternative form of the flagellin can survive because the host doesn't have an immune response against them. So this host immune response to one flagellin selects for those expressing the other form of the flagellin. And only one type of flagellin can be expressed at one time. Never both, never neither. And this is due not to slip strand mispairing. So slip strand mispairing drove that process of phase variation. The process that's driving this specific case of allelic variation or antigenic variation is inversion. So a piece of DNA can literally be flipped around in the bacterium. And because it's near the end of the lecture and it's Friday and everyone's taking exams, I'm not going to try and cram this into the last 3 minutes because this is kind of complicated. So we'll deal with this on Monday. Good luck on the exams for those of you who haven't taken it yet. And have a great weekend. Hey!
Hey, I got your text.